can you describe how COVID vaccines work for someone who maybe has never taken biology?
So for the viruses, they need several mechanisms to enter the cell. So in particular for the SARS-CoV-2, which the pathogen caused COVID-19, they use the spike protein called the S gene, or the S protein, spike protein, which is kind of similar to like a cron. That's why we call it coronavirus. So that spike protein can be recognized, and can further bind to the receptor. Receptor means, on the cell surface, there's certain kind of a small molecule or kind of a protein that can recognize this spike protein. And once they attach to the receptor called the ACE2 on the target cell, then can initiate the kind of a special biochemical processes, and the SARS-CoV-2 virion will gradually get into the host cells, and then can release the cargo, like a genetic information, into the host cell. So yeah, so the spike protein is very critical, as I have described here. It is responsible for how efficient or what kind of cell that the SARS-CoV-2 can get into the target cell, get into the host. For the vaccine, we generally tried, what we did is to introduce, where we deliver, where we put the pathogens, a piece of the protein or piece of the the immunogen, what we call protein, into the host cell. The idea is, once you put this in the foreign piece of it, the immunogen or the proteins into the host cell, because it is foreign, then your host, or the patients or the participants' immune system will recognize the foreign stuff, and they will try to target it and disrupt it or destroy it. So during this process, they will initiate an array of different tools. So the tools means the different the antibody. So antibody is kind of special, like a protein produced by your body that can bind to that foreign stuff and help to remove that foreign stuff. So that's from your, so that antibody can be in your blood or in your mucosal fluid. So just apart from that, we also have several other cellular tools. For example, like your red blood cell, there are kind of special cells called the T cell. That cell can also help to remove the foreign stuff that get into your cell. So in both ways, either from this antibody or from the T cell, our host body is trying to develop this kind of special this special tools to get rid of those. So what our vaccine is trying to do is just deliver this kind of special foreign antigens into our body, and try our best to produce these different antibodies, the T cells, and once we have that one, so for a long time, if we have always have those different tools available, we're ready for the second exposure or from the real virus infection. So in that way, because you're already immune to it, so you're already ready for, you're well prepared to have those antibody and T cells. So during the second infection, so your antibody can directly target the incoming the pathogens, and directly can either just eliminate it, or just try to minimize the exposure of our cells to that invasion, or even they get into the cell. Because if you have T cell immune responses, you have antibodies, it will make your disease is less severe. So that's how the vaccine, from the mechanistic inside, how the vaccine trying to do. But meanwhile, the best thing is that the vaccine will not cause the disease as the real virus infection did. So in that way, they will minimize the side effects, but try to induce those immune responses in your body that try to help you.
When an antibody binds the spike protein cannot bind a human host cell, so the virus cannot enter the human cell anymore. Is that
right? So one of the function for the antibody is try to bind this very sensitive epitope where a very sensitive region on the spike proteins. So once they bind, then the spike protein cannot bind to the receptor ACE2 on the cell. Therefore, they can block the virus infection. So that's one of the mechanisms how the antibody can inhibit viral infection.
You said that these vaccines, they try to mimic germs. Like, could you sort of describe how the different vaccines mimic the germ in different methods or in different ways?
Yeah. So basically, different types of vaccines has unique features, and they can confer the protection in different ways. But the core idea is they are trying to deliver the key, like we call the antigen, or a piece of the the proteins or the nucleic acid from the pathogens. So that is, they try to deliver the pathogen into the host cells and use that immunogen to trigger the immune responses in the host cells. So different type of vaccines have different methods. For example, traditionally people are trying to, where the scientists trying to deliver the antigen itself. For example, by deliver the protein subunit into the host. So those are the very traditional way. For right now, there are several companies such as the Novavax are developing the vaccines based on that platforms. And most recently, there are advanced several newly novel vaccine platforms. So these are they're not trying to deliver the antigen itself, but instead they're trying to deliver the what we call the nucleic acid, or what we call the genetic information, or in other words, called a blueprint, into the host, and the blueprint or the genetic information will instruct the host cells to express the antigens, and therefore the antigen can further trigger the immune responses. So those new technologies will include right now we have a mRNA vaccine which was developed by the Moderna and Pfizer. So they're very helpful and immunogenic right now, and has been approved by the FDA. And for us in particular, we are collaborating with Johnson & Johnson. We also utilize a novel technology called a viral vector vaccine. So for that one, we use the virus. Actually, there's another virus, not SARS-CoV-2, not the virus caused the COVID-19, but another virus that generally they cause a very mild cold, like the flu-like symptoms or kind of common colds. We call it adenovirus. In particular, we call it Adenovirus 26. It is kind of subtype. So that virus is a kind of has been modified. They remove the essential virulent genes and make the virus can only infect the target cell or infect the host cell, but does not cause any illness. So we insert the immunogen, or what we call the piece of the spike protein from the SARS-CoV-2 into the advector, and then allow the advector to bring that piece of information into the host cell. Therefore, can express the SARS-CoV-2 spike protein there and further induce immune responses. So that's our technology, which is in cooperation with Johnson & Johnson, and they're now has been approved as the third vaccine in the US.
For the Johnson & Johnson vaccine specifically, could you also describe again how the adenovirus vector presents the antigen?
So for the Johnson & Johnson adenovirus, so for that one we insert the SARS-CoV-2. SARS-CoV-2 is the viral pathogen that caused the COVID-19. So we specifically took the one piece of the genes, or one piece of the genetic information from the SARS-CoV-2, and we call the spike protein gene, so spike gene. So we put that gene, insert into another, the virus that causes common cold virus, common cold. So that virus called Adenovirus 26. So we put that SARS-CoV-2 spike gene into the Ad26 common cold virus, just to make this chimeric the virus. And this chimeric virus we call the viral vector vaccine, and they have a unique property. So that for that vector, we remove some of the virulent genes. Virulent means you delete some of the genes that keep the virus, there are some genes that are responsible to cause disease. For the common cold virus, for example, the Adeno26. If you remove that genes, the virus cannot cause disease anymore. It is become safe right now. So by using that modified Ad26 vector, we insert the spike genes into the vector and make this chimeric virus. And the virus can infect. Once you take the shot, then the virus can deliver the spike gene into the host cells and start to express those proteins, these foreign proteins, and then induce this immune response to produce the antibody and T cells. But because it is modified, the virus itself cannot multiply. It is they call it replication incompetent. So eventually the virus will gone because they cannot replicate. They will be cleared by your host by the host immune systems. But it is immunogenic. It can trigger immune responses, can induce this antibody and T cells, and eventually can be protective against the SARS-CoV-2 infection.